Hello students, I am Priya Lagwankar. Today I have come up with an interesting topic, and that is bacteria and viruses. Students, you know that both bacteria and viruses, they come under the category of microorganisms. Microorganisms, of course, which cannot be seen by naked eyes. So today we are going to know more about these bacteria and viruses. Now students, in front of you there are various types of bacteria. See, these types of bacteria, they are based on their shapes. So first one is circular shape, students. Now the circular shape bacteria, when they are in pairs, such bacteria, they are called as diplo. Okay. Then next are strepto, where the circular shape bacteria, they are forming long chains. And staphylo, when the bacteria, they are forming clusters. That means circular bacteria, they are called as diplococci, streptococci or staphylococci, like that. Now next is rod shaped bacteria. See, the rod shaped bacteria which are oval, they are called as cocobacilli. Then the ones which are elongated, rod like, so these are streptobacteria, and further elongated, they are mycobacteria. Here curved forms of bacteria, they are there. Vibrio, when they are curved rod like. Then spiral shape, they are spirilla. Okay, and further elongated spiral ones, they are called as spirochetes. Like that. Sometimes the bacteria can be even of other shapes like helical or club shaped, or here elongated thread like. So depending on the shapes of bacteria, these are the types. Have you understood? For example, the cholera bacteria, they are called as Vibrio cholerae. That means the bacteria causing cholera, they are of Vibrio shape. The shape of bacteria is this. Means depending on the shape of bacteria, they are named. Okay. Students, we have already seen that the bacteria, they come under prokaryotes. Okay, they are of course unicellular, and that cell is a prokaryotic cell. Now here in this diagram you can see how the bacterium, it multiplies, or how one single bacterium divides into two. Here you can see first the inner material, which is called as nucleoid. That material is divided into two parts, and a notch is formed in the cell wall of bacterium. And now here finally a new cell wall is generated, and the two bacteria finally they are separating from each other. Here in this video you can see actually how they are multiplying and how fast it is occurring. The bacteria, they can divide very rapidly, and that is why their number can increase very fast. Students, as you can see here, one bacterium, it is dividing into two. So likewise in this video, see why their number is increasing so rapidly. Of course, a single bacterium, it is dividing into two. Then two, further they are dividing into two. That means four are formed. Then those four again dividing and forming 8 bacteria. And 8 by division, they are forming 16. So likewise, the number is increasing by a cumulative effect. And that is why so rapid rise in their number can be seen. Students, now there is curd in front of you. You know, when we have to convert milk into curd, we have to add a small amount of curd into it. That curd which we are adding is actually nothing but a culture of lactobacilli. Do you know, students, that contains lactobacilli, and those lactobacilli, they convert milk into curds. And that is why when we have to convert milk into curd, we have to add curd into it, which contains those lactobacilli. They bring about certain chemical process, and they bring about curdling of milk. So students, when we are adding curd to the milk sample, of course the bacteria, the lactobacilli present in that curd sample, they multiply and they convert the entire milk into curds. So this process is brought about with the help of bacteria. That means in dairy products, the bacteria are playing a very important role. Students, you can see here in this particular picture, these are the roots of leguminous plants. Leguminous plants means the plants which are having such legumes or pods. So in such leguminous plants, the root bear such rounded nodules. What these nodules are? See, these nodules are formed by a bacteria called as rhizobium bacteria. These bacteria, they stay inside the nodules formed on the roots of such plants, and what they do, they fix the atmospheric nitrogen. Now, what is meant by fixing of nitrogen? There is a large amount of nitrogen in the atmosphere around us, but we cannot take up that nitrogen directly. Not we, even plants cannot take up that nitrogen directly. But nitrogen is a very important component of our body proteins. We all living things require nitrogen, but we cannot take it from the atmosphere. Then how it enters the living organism's body? See, it is due to presence of these bacteria. What they do, they can take up the atmospheric nitrogen and they can convert it into nitrogen compounds. And when they are growing on these plants, they transfer those compounds to the plant. And of course, the plant uses it for formation of proteins. When such plants, they are consumed by animals, they come in the food chain. So that is a very great benefit of these bacteria. Some other kind of nitrogen fixing bacteria, they are also present in the soil, and they also do the same procedure, and they also convert the atmospheric nitrogen into nitrogen compounds, and they are made available to all living organisms through plants. Now students, here in this photograph you can see the process of tanning of leather. Now what exactly it is? You know that the skin of dead animals is converted into leather. But while converting that skin into leather, some processes have to be made. And in those processes, in the first step, to remove the degradable part of the skin, that skin is treated with bacteria. So bacteria are playing important role in such processes like tanning of leather. Students, now you can see antibiotics in this particular picture. These antibiotics are nothing but chemicals formed by some bacteria and some fungi. And these chemicals are capable of killing other kind of bacteria. That means they are the products formed by some fungi or some bacteria, and they are killing some other type of bacteria. This property of these chemicals is used to kill the disease causing bacteria. See students, the harmful bacteria, they are treated using antibiotics. But basically in the formation of antibiotics, bacteria are only used. Means they are playing a very important role in manufacturing of antibiotics. Here you can see the agrobacteria in this particular picture. Now these agrobacteria, they are invading, they can infect the plant cell, and that bacterium, while infecting the plant cell, can incorporate its piece of its DNA into the plant DNA. So what is done, students? Whatever desired gene is there, that gene is first introduced into this particular bacterium. See, this is the gene introduced in this bacterium using biotechnology, and then when this bacterium, it infects the plant cell, that gene can be transferred to the plant cell and it gets incorporated in the DNA of that plant cell. That means that plant cell now can show the desired characters expressed by that particular gene. So that is how the gene has been introduced into the plant cell using these particular bacteria. So they are acting as tools in the biotechnology. In this way, there are various uses of bacteria. You know that some bacteria, they are present in our gut also, they are present in our digestive system also, and they are helping in the process of digestion. They are helping in conversion of undigested food into feces. Similarly, students, you know, bacteria, they bring about the decomposition of organic matter. They are used for conversion of organic matter into manure. That means they are used for composting process. There are certain bacteria which are being used to remove the oil spills on the seawater. Likewise, there are many uses of bacteria. So far we have seen the uses of bacteria. But are all bacteria useful? Of course not. Some bacteria, they are very useful to human beings. They are quite beneficial. But at the same time, some bacteria, they are causing various diseases in human beings. Not only in human beings, some other kind of bacteria, they are causing diseases in animals and plants also. So let's see first what are the different diseases caused by bacteria in human beings. See here in this particular picture, you can see the different diseases like typhoid, cholera or pneumonia, diphtheria, tuberculosis, tetanus. You must have heard all these names. So here whatever diseases are shown, those diseases are caused by bacteria. These are all bacterial diseases, students. And of course, as we have already seen the antibiotics, all these diseases, they are treated using antibiotics. Okay, so that means antibiotics formation is also by bacteria and they are being used also against bacteria. As I told you, the bacteria are causing diseases not only in human beings, they are causing diseases in plants also. So here you can see different plants affected by different bacterial diseases. Apart from diseases, some bacteria, they even cause spoilage of food or spoilage of some other articles, and they can even cause food poisoning. So likewise, as some bacteria are very useful, at the same time some bacteria are quite harmful. And there is a third category of bacteria also, that is neither useful nor harmful. Students, so far we have seen that some bacteria, they are harmful, some are useful. Now let's see how exactly the inner structure of a bacterium is. The bacterial cell is a prokaryotic cell. It contains the genetic material, that is DNA, floating in the cytoplasm. See, this is the cytoplasm. This is the DNA, also called as nucleoid, and there is plasma membrane and cell wall around the cytoplasm. Okay, and here this particular bacterium, it is showing presence of hairy structures, small hairy structures, which are for movement and which are for attachment to any substratum. And here there is flagellum also for the locomotion. With the help of flagella, they can move. In addition to this nucleoid, there is a small circular DNA, and that is called as plasmid. Means this nucleoid and these plasmid, they are two different things. This is the smaller one and this is called as plasmid. Plasmid is shown by certain bacteria. Then there are ribosomes in the cytoplasm, and of course they are playing a role in the synthesis of proteins. So that is the general structure of a bacterial cell. Students, now let's see what is the structure of a virus. Here you can see that there is a central DNA molecule. See, other living things, they have DNA only as their genetic material, but only viruses are there which can have DNA or RNA molecule as their genetic material. Now this DNA is encircled with a protein coat, then further outer protein coat is there. Like, means you can say a thread kept in a box, and that box is again surrounded by a ball, and then there are pins inserted in the ball. So likewise the structure is there. So here also these are also proteins, and this lipid envelope around that, and there are envelope proteins inserted in that lipid envelope. So that is the general structure of a virus. Now students, the bacteria, they are capable of surviving independently, and we have also seen how they divide, how they multiply. The bacteria, they can survive independently. They are prokaryotes. Then are we going to call virus also as prokaryotes? No students. Viruses are not even prokaryotes because they are considered to be on the boundary line of living and non-living things. Why I am saying so? See, because students, when the virus is outside the body of any other organism, it is like a non-living particle. No life processes are going on in the body of virus when it is outside the body of any other organism. But when it enters the body of any other living thing, then only it can multiply. We are going to see it further. But remember, the viruses are considered to be on the boundary line of living and non-living things, and they are not even classified as prokaryotes. Bacteria are prokaryotes, but viruses are not prokaryotes. See, the viruses are of different types. Some they invade the plant cells, some they enter the animal cells, and some of them they can even enter the bacterial cells. So when the virus, it enters inside any other living cell, what happens? See, the nuclear material of the virus, it is released inside the cell, and then using the mechanism of the host cell, see, the virus is releasing the nuclear material inside it. The mechanism or the material from the host cell is being used, and the viruses, they are multiplying themselves. Here you can see it clearly, students. See, the bacterium has been invaded, the bacterial cell or any other cell. When it is invaded by the virus, what happens? The virus is releasing only genetic material inside. The capsule is remaining outside. Then that viral genome, it is forming its copies. They are multiplying. Not only that, the spare parts of virus, they are also synthesized from the proteins present in the cell, and that is how new viruses are manufactured inside the cell. Then they destroy the cell, and then they are released outside. So that is how the viruses, they multiply. That means the viruses, they cannot multiply on their own. They need presence of host cell only. They cannot show any life processes, or they cannot even multiply outside the host cell. Now students, there are different types of viruses in front of you. The viruses are basically categorized into three types. The viruses infecting plants, the viruses infecting animals, and the viruses which infect the bacteria. The viruses which infect the bacteria, they are called as bacteriophage. So they are having a particular rocket-like shape. Okay, and here you can see the different viruses and their different shapes. The viruses, they are causing different diseases in human beings. Okay, like chicken pox or measles or hepatitis or even common cold is caused by viral disease. Students, the disaster which we are suffering from at present, and that is COVID-19 disaster, and of course cause of this disease is also a virus, and that is coronavirus. Even diseases like rabies or AIDS, they are also caused by viruses. So students, you find out which different diseases in human beings, they are caused by viruses, and which different diseases in human beings, they are caused by bacteria. So the viruses which infect the plants. Of course, they are responsible for causing some disease in those plants also. So here you can see in this picture the plants affected with viral diseases. Students, so far we have talked about the bacteria and viruses. Now, what is their size exactly? Students, in this particular picture, you can see what is the comparative size of a bacterium and a virus. You know that the units micrometer or micron and nanometer, they are used to measure the microscopic things. We know that when 1 millimeter is divided into 1000 equal parts, each part is called as 1 micrometer, and when 1 micrometer is further divided into 1000 equal parts, each part is called as a nanometer. In this picture you can see this is the human RBC. That means red cell present in our blood. See the E. coli bacterium, the comparative size of E. coli bacterium and the RBC. So the RBC is much more larger than a bacterium. And here you can see a bacterium. It's much more larger than the viruses. See, the viruses, their sizes are in nanometers. Here you can see they are in nanometers, whereas the bacteria is measurable in micrometers. So bacterial cell is comparatively much larger than the virus, and it is much smaller as compared to human RBC. So students, in short, while talking about the size of bacteria and viruses, the smallest bacterium is bigger than the largest virus. Okay, the bacteria which is the smallest one, it is comparatively bigger than the biggest virus. That means bacteria are much more bigger in size than the viruses. So that is one difference in bacteria and viruses, the difference of size. We have also seen their structures. So you can compare their structures and find out the similarities and differences in them. One more important point, bacteria, they can be useful or harmful or neutral, but all viruses, they are always harmful. They are causing disease in some or other kind of organisms. But both bacteria and viruses, they are being used in biotechnology as tools for introduction of genes in the plant cell or animal cell and get the desired effects. So students, you must have found this information about bacteria and viruses interesting. You must be reading, you must be watching the videos. So all the best for your further journey. Today we are going to stop here. Goodbye students.